Hello and welcome to class. This is Biochemistry BCH 302 for the College of Natural Health Sciences, Bermuda. I will be your professor, Dr. Delsina B. Burrows, and welcome to lecture number 1. Now, I do realize for some of you, you may not have had any extensive chemistry or biology either in high school or maybe even in primary school. You may not have had any exposure to biology or chemistry or even physics for that matter. Well, I don't want you to be intimidated by the name of the course as well as the things that we will be looking at in this class. Yes, biochemistry can be pretty involved. However, we will do our very best to break down the information so that it is understandable and for you as natural health care practitioners to have the very basic information that you need to be a successful natural health practitioner. You're not studying to be a biochemist, so that means that the amount of detail that you'll have to have going into this will be different than what you're actually presently studying, which is to become a natural health practitioner. But we will certainly give you a very strong and a firm foundation for which to launch your practice from, and for you to be an evidence-based, science-based practitioner, you do need to have an understanding of biochemistry. But as I said, this is by no means going to be an intimidating course. It will be very, very manageable. You will understand the broader concepts, which is all that is really needed in order for you to be a successful natural health practitioner. The course objectives for Biochemistry 302 are as follows. You'll be able to explain the elements, atoms, ions, chemical bonding, states of matter, gas behavior, chemical reactions and equilibrium, and the importance of energy transfer, catalysts, and identify applications in biological systems. You'll be able to identify functional groups and analyze chemical nature of all organic molecules, including carbohydrates, lipids, proteins, and nucleic acids. You'll be able to explain common chemical reactions and properties associated with organic functional groups and state their applications in biological systems. You'll also be able to describe the function and main biochemical pathways for each major biochemical group. You'll be able to apply the knowledge of the major biochemical pathways and how they relate to health and disease states. And you'll be able to illustrate regulation and integration within the main biochemical pathways and explain how this contributes to normal homeostasis in the body. The biochemistry course is structured in 3 parts. First, the theory. And for the theory, you will receive that in a series of 12 video lectures over the course of the semester. Also, we have what is referred to as a theoretical lab. And the theoretical lab is based on assignments that will come from the Essential Biochemistry textbook, which I'm going to talk about in just a little bit. And also some videos that are on YouTube that you can watch in accompaniment of doing the assignments. And then we also have what's referred to as a lab practical, and this is a 10-week biochemistry home experiment that you're going to perform. And I'm going to give you some more information about the lab practical in a separate video. But it's really nothing to stress about. Once again, just something very, very simple that you'll be able to do at home, and you're going to track the progress of it over a period of 10 weeks. Okay, so like I said, I'll give you more information about that in a separate presentation. Now, in terms of the course textbooks and reference materials, you will have lecture notes, and for each of the 12 videos you will automatically get a series of lecture notes in the form of PDFs that will help you to go through the material. So you'll obviously be able to see the videos, but you will also have the backups to the videos in the form of the PDF notes. And in addition to that, you will also have access to the Pratt and Cornely's Essential Biochemistry textbook. And for each chapter that is assigned for you to read, you will automatically get the lecture note PDFs for that. So you don't have to worry about purchasing the Essential Biochemistry textbook unless you really want to. And then, of course, the University of Texas at Austin, they have a YouTube channel entitled Fundamentals of Biochemistry. And what I will do is I will probably post the link to each corresponding chapter of the Essential Biochemistry textbook chapter, whichever chapter we're looking at for that particular week. I'll go ahead and I'll post the YouTube video that corresponds with each of those chapters. So if you would like to look at the video while you're also looking at the Essential Biochemistry notes, then it'll help to reinforce the information. But for this particular class, you do not have to purchase any textbook because you will be given all the lecture notes that you will need to have in order to complete the course. Now, for today's lecture objectives, at the end of this lecture, students will be able to provide an operational definition for biochemistry. They'll be able to state why it is important for natural health practitioners to have a thorough understanding of biochemical processes. You'll be able to identify the four main classes of biomolecules, and you'll be able to describe the relationship between biochemistry, genetics, and molecular biology. So let's start with the definition of what biochemistry is. Biochemistry is a branch of science that explores the chemical processes that exist within and are related to living organisms. Biochemistry is a laboratory-based science that combines both biology and chemistry. The main focus of biochemistry is on the processes that occur at the molecular level. That is, things that are happening inside the cells. Biochemistry also studies components such as proteins, lipids, organelles, and how cells communicate with each other. The primary focus of biochemistry is to understand how biological molecules lead to the processes that occur within living cells, which is, in turn, related to the study and understanding of tissues, organs, and organisms as a whole. So biochemistry seeks to understand the relationship between function and structure. A large part of biochemistry is concerned with the structures, functions, and interactions that exist amongst biological macromolecules, such as proteins, nucleic acids, carbohydrates, and lipids, which provides the structure of cells and perform many of the functions that are associated with life. So we will begin our study of biochemistry by first considering cell chemistry. The chemistry of the cells also depends on the reactions of smaller molecules and ions. These can either be inorganic, as in water and metal ions, or organic, such as the amino acids that are used to synthesize proteins. Let us now consider metabolism and biochemical research. The mechanisms through which cells harness energy from their environment via chemical reactions are referred to as metabolism. The discoveries of biochemistry research are applied primarily in the fields of medicine, natural health practice, nutrition, and agriculture. In medicine, biochemists examine the causes and cures of diseases. In nutrition, the effects of nutritional deficiencies are studied in order to determine how to preserve health and wellness. In agriculture, biochemists investigate soil and fertilizers and look for ways to improve crop cultivation, crop storage, and pest control. So why, you may ask, is it important for osteopaths and naturopaths to understand biochemistry? Well, osteopaths and naturopaths need to have an understanding of how the structure of a molecule is related to its function. This will in turn allow the practitioner to forecast how molecules will interact and respond to natural and/or manual interventions. Natural health practice is based on sound scientific body of knowledge that is evidence-based. Intervention is guided by what we know about living organisms. So now let us consider the chemical elements. About 24 out of 92 naturally occurring chemical elements are vital to various types of biological life. While most organisms share element needs, there are a few differences between plants and animals. For example, ocean algae use bromine, while land plants and animals don't seem to need it. Just 6 elements, which are carbon, hydrogen, nitrogen, oxygen, calcium, and phosphorus, make up 99% of the mass of living cells, including those in the human body. In addition to the 6 major elements that make up most of the human body, humans require smaller amounts of possibly 18 or more. So here we have the periodic table of elements, which is all the known elements that appear in the universe. We look at them according to the biological elements tier system. In the first tier, these are the most abundant elements that can be found, beginning with carbon, nitrogen, hydrogen, and oxygen. The second tier means that they are less abundant than the first, and these would include calcium, potassium, kalium, sulfur, chlorine, sodium, and magnesium. The third and fourth tiers are trace elements, and transitional metals, iron, and manganese are amongst this group. This relative abundance is the same for all life forms, a small subset of the full periodic table. Let us now discuss biomolecules. The main 4 classes of molecules in biochemistry, which are often referred to as biomolecules, are carbohydrates, lipids, proteins, and nucleic acids. Many biological molecules are polymers. A polymer is a chemical compound where molecules are bonded together in long repeating chains. Monomers and polymers. All living things are made up of the same building blocks. Monomers are basic structural units that are smaller units from which larger molecules are made. Polymers, also referred to as macromolecules, are formed when monomers join together. Condensation joins 2 molecules together and involves the elimination of a water molecule. Hydrolysis breaks a chemical bond between 2 molecules and involves the use of a water molecule. Let us now consider carbohydrate molecules. The function of carbohydrates includes the storage of energy and provision of structure. Sugars are categorized as carbohydrates, but not all carbohydrates are sugars. Carbohydrates are used to store energy and genetic information, while also playing an important role in cell interactions and communications. The simplest type of carbohydrate is a monosaccharide, which contains properties like carbon, hydrogen, and oxygen, mostly in the ratio of 1 to 2 to 1. Glucose is one of the most vital carbohydrates. Other types include fructose, sugar, commonly associated with the sweet taste of fruits, and deoxyribose. A monosaccharide can switch from acyclic open chain form to a cyclic form through a nucleophilic addition reaction between the carbonyl group and one of the hydroxyls of the same molecule. The reaction creates a ring of carbon atoms closed by one bridging oxygen atom. The resulting molecule has a hemacetal or hemaketal group, depending on whether the linear form was an aldose or a ketose. The reaction is easily reversed, going back to the original open chain form. When it is in its cyclical form, the ring is usually made up of 5 or 6 atoms. These forms are called furanoses and pyranoses, respectively. For example, the aldohexose glucose can form a hemacetal linkage between the hydroxyl on carbon 1 and the oxygen on carbon 4, creating a molecule that is a 5-member ring called glucofuranose. When two monosaccharides go through the dehydration synthesis, where a molecule of water is released because 2 hydrogen atoms and 1 oxygen atom are lost from the 2 monosaccharides, the new molecule, which is made up of 2 monosaccharides, is referred to as a disaccharide and is joined together by a glycosidic or ether bond. A reverse reaction can also occur where a molecule of water is used to divide a disaccharide and break the glycosidic bond. This is referred to as hydrolysis. When 3 to 6 monosaccharides are combined, it is referred to as an oligosaccharide. Numerous monosaccharides combined together make a polysaccharide. Sugar can be categorized as having reducing or non-reducing ends. A reducing end of a carbohydrate is a carbon atom that can be in equilibrium with the open chain aldehyde, which is aldose, or keto form, which is ketose. Lactose has a reducing end at its glucose moiety, while the galactose moiety forms a full acetal with the C4OH group of glucose. The next biomolecule that we will consider is lipids. Lipids are made up of a varied range of molecules and to some extent is a blanket for relatively water-insoluble or nonpolar compounds of biological origin, and this includes fatty acids, fatty acid-derived phospholipids, sphingolipids, waxes, glycolipids, and terpenoids. Some lipids are linear aliphatic molecules, whereas others have ring structures. Usually, lipids are made from one molecule of glycerol joined with other molecules. In triglycerides, which is the main group of bulk lipids, there is one molecule of glycerol and 3 fatty acids. In that case, fatty acids are considered to be the monomer and may be saturated, which means they have no double bonds in the carbon chain, or unsaturated, which means that they have one or more double bonds in the carbon chain. A great deal of lipids have a certain polar character in addition to being mostly nonpolar. Overall, the greater part of their structure is nonpolar or hydrophobic, which means that it does not interact well with polar solvents such as water. Another part of their structure is, however, polar or hydrophilic, and it will incline itself to associate with polar solvents such as water. This makes lipids amphiphilic molecules, meaning that they have both hydrophobic and hydrophilic portions. When it comes to cholesterol, the polar group is a simple OH, which is hydroxyl or alcohol. In the case of phospholipids, the polar groups are significantly larger. Lipids are an essential part of the human daily diet. Most oils and milk products that are used for cooking and eating are made up of fats. Foods that contain lipids undergo digestion inside the body and are broken down into fatty acids and glycerol, which are the final products of fats and lipids. The next biomolecule that we will consider is the protein molecule. Proteins are very large molecules, referred to as macrobiopolymers, that are made from monomers referred to as amino acids. An amino acid comprises of a carbon atom which is bound to 4 groups. One group is an amino group, NH2, and one is a carboxylic acid group, referred to as COOH, although these exist as NH3 plus COO under physiologic conditions. The third is a simple hydrogen atom, and the fourth usually denoted as R, and this can be different from each amino acid. There are 20 standard amino acids. Each contains a carboxyl group, an amino group, and a side chain referred to as R group. What makes each amino acid different is the R group, and the properties of the side chains significantly influence the overall three-dimensional conformation of a protein. Some amino acids have functions that can be undertaken by themselves or in modified form. Amino acids can be joined by a peptide bond. In this dehydration synthesis, a water molecule is removed and the peptide bond joins the nitrogen of one amino acid's group to the carbon of the other's carboxylic acid group. The resultant molecule is referred to as a dipeptide, while short chains of amino acids, typically less than 30, are referred to as peptides or polypeptides. Longer chains merit the name proteins. There are some proteins that perform largely structural roles. For example, the movements of proteins actin and myosin are ultimately responsible for the contraction of skeletal muscle. One basic attribute that many proteins have is that they specifically bind to a specific molecule or class of molecules. Proteins can be extremely selective in what they bind. A good example are antibodies. They attach to one specific type of molecule. The most important proteins are the enzymes, as virtually every reaction in a living cell needs an enzyme to lower the activation energy of the reaction. These molecules recognize precise reactant molecules called substrates. They then catalyze the reaction between them. When the activation energy is lowered, the enzyme speeds up that reaction by a rate of 1011 or more. A reaction that would typically take up to 3,000 years to complete freely might take less than a second with an enzyme. The enzyme, however, is not used up in the process, but is free to catalyze the same reaction with a new set of substrates. Using numerous modifiers, the activity of the enzyme can be regulated, aiding control of the biochemistry of the cell as a whole. The structure of proteins is described in a hierarchy of 4 levels. The primary structure of a protein basically consists of its linear sequence of amino acids. The secondary structure of a protein is concerned with local morphology. Some combinations of amino acids will curl up in a coil referred to as an alpha helix, or into a sheet called a beta sheet. The tertiary structure of protein is the entire three-dimensional shape of the protein. This shape is determined by the arrangement of amino acids. Lastly, the quaternary structure is concerned with the structure of a protein with several peptide subunits. An example is the hemoglobin, which has 4 subunits. Not all proteins have more than 1 subunit. Consumed proteins are typically broken down into a single amino acid or dipeptides in the small intestine and then absorbed. Intermediary products of glycolysis, the citric acid cycle, and the pentose-phosphorus pathway can be used to create all 20 amino acids, although most bacteria and plants have all the essential enzymes to synthesize them. Humans and other mammals, however, can synthesize only half of them. Humans and other mammals cannot synthesize leucine, lysine, methionine, phenylalanine, isoleucine, threonine, tryptophan, and valine. All of the aforementioned are the essential amino acids as it is essential to ingest them. Mammals do not have the enzymes to synthesize asparagine, aspartate, cysteine, glutamate, glutamine, glycine, alanine, proline, serine, and tyrosine, the non-essential amino acids. Even though they can synthesize arginine and histidine, they cannot produce it in sufficient quantities for young, growing animals, and so these are often considered essential amino acids. When the amino acid group is detached from an amino acid, it leaves behind a carbon skeleton referred to as an alpha keto acid. Enzymes referred to as transminases can easily move the amino group from one amino acid, creating an alpha keto acid, to another alpha keto acid, creating an amino acid. This is vital in the biosynthesis of amino acids, as for a variety of the pathways, intermediates from one biochemical pathways are transformed into the alpha keto acid skeleton, and then an amino acid is added, most times through transamination. The amino acids can then be linked together to create a protein. To determine whether two proteins are related or homologous, scientists use sequence comparison techniques. Techniques like sequence alignments and structural alignments, are great tools that help scientists detect homologies between related molecules. The importance of discovering homologies amongst proteins goes beyond creating an evolutionary pattern of protein families. By discovering how alike two protein sequences are, we often obtain knowledge about their structure and consequently their function. Now let's consider the fourth and final group of the biomolecules, nucleic acids. Nucleic acids are complex, high molecular weight biochemical macromolecules that can carry genetic information in all living cells and viruses. The monomers are referred to as nucleotides, and each is made up of three components: a nitrogenous heterocyclic base, it can be a purine or a pyrimidine, a pentose sugar, and a phosphate group. The most common types of nucleic acids are deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. The phosphate group and sugar of each nucleotide bond with each other to create the backbone of the nucleic acid, while the sequence of nitrogenous bases stores up the information. The most common nitrogenous bases are cytosine, guanine, thymine, adenine, and uracil. The nitrogenous bases of each strand of a nucleic acid will form hydrogen bonds with other specific nitrogenous bases in a complementary strand of nucleic acid. Adenine binds with thymine, and uracil, thymine binds only with adenine, and cytosine and guanine can only bind with one another. Aside from being the genetic material of the cell, nucleic acids often play a role as second messengers. They can also form the base molecule of adenosine triphosphate, which is also referred to as ATP, the primary energy carrier molecule found in all living organisms. Also, the nitrogenous bases possible in the two nucleic acids are different. Adenine, cytosine, and guanine occur in both RNA and DNA, while thymine occurs only in DNA and uracil occurs only in RNA. So now let's talk about the relationship between biochemistry, genetics, and molecular biology. Biochemistry researchers employ the use of specific techniques that are native to biochemistry, but gradually combine these with techniques and ideas that are developed in the fields of genetics, molecular biology, and biophysics. There has never been a division amongst these disciplines in terms of the content and methodology, hence biochemistry. So to summarize some key points of today's lecture, biochemistry is the study of the chemical substances and essential processes that occur in living organisms. Biochemists focus largely on the role, function, and structure of biomolecules. Genetics is the study of the effects of genetic differences in living organisms. Molecular biology is the study of molecular foundations of the process of replication, transcription, and translation of genetic material. The aim of chemical biology is to develop new tools constructed on small molecules that allow slight perturbation of biological systems while providing thorough information about their function. Okay, so for assignment number 1, there are 2 parts to this. The first, in an essay of no less than 200 words, you're going to provide an operational definition for biochemistry. You're going to describe why it is important for natural health practitioners to have an understanding of biochemistry as it relates to humans. And you're going to provide a description of the 4 main biomolecules. In the second part of the assignment, you're going to read the Bioenergetics Open Access article on that should be mitochondria evolution, and you're going to discuss its significance in biochemistry advances. And this could be 100 words. And as always, if you need to contact me about any of the content of today's lecture, you can shoot me an email, or you can give me a call on my cell phone, 703-7327. And my office hours are, as always, on Thursdays from 4 to 6 p.m. And again, as I mentioned earlier, I don't want you to get too bogged down with some of the minute details with respect to biochemistry, because biochemistry can get pretty involved. But for our purposes, it's more important that we understand concepts, the broader concepts that biochemistry is a thing that we are concerned about, and we need to have that broad-based working knowledge of biochemistry as it relates to our practice as natural health care practitioners. So don't get too bogged down with some of the minute details, but more the broader concepts. And that's the reason why the assignments are laid out the way they are, for you to provide a description and have that working understanding of what it is as you go forward. And a lot of this will start to make more sense as you do some of your other studies, when you study nutrition and get further into your anatomy and physiology, and also some of the techniques that we use to treat our clients. This will all start to come together, okay? All right, so with all of that, have a great week, and I look forward to also seeing your responses in the forum for this week, which will be on mitochondria evolution. So be on the lookout for that, okay? Do take care.